Hello everyone! I was watching my brother play this video game and he used some sort of a cheat code that let his characters tunnel through walls. Imagine you can do that in real life, and it turns out you can at a quantum level. And more importantly, it can happen in our DNA and can be a cause for mutations that lead to the second leading cause of death, cancer. An estimated 2 in every 5 Canadians will be diagnosed with cancer throughout their lifetime, and approximately 1 in every 4 will die from cancer. Therefore, as a grade 11 student attending Westdale Secondary School, and as a proud Canadian citizen, I have purposed my science fair project to research how quantum tunneling can induce mutations in our DNA that culminate into cancerous cells. DNA is an organic molecule with a molecular structure that contains a genetic code which guides living things to develop, live, and reproduce. Within the blueprint of life, spontaneous spelling mistakes or mutations can occur, leading to potentially catastrophic errors. One possible candidate for these spontaneous mutations is the quantum mechanical tunneling of hydrogen atoms within the bonds. DNA is made up of molecules called nucleotides. Nucleotides pair together in a specific way called the base pairing rule. Watson and Crick proposed a concept of DNA as a double helix in 1953. In DNA, adenine always pairs with thymine, and cytosine always pairs with guanine. These nucleotide bases are paired through hydrogen bonds and are linked to the sugar phosphate chains that comprise the double helix's 2 backbone strands. The hydrogen bond is essentially a hydrogen atom that holds some molecules together, a single proton shared between 2 separate electrons. It's not too strong of a bond. It's strong enough for molecules to fit together to make very complicated structures like proteins and enzymes, but it's not too strong of a bond that it can't be broken easily enough so that these molecules can rearrange themselves. The figure on the left represents the adenine-thymine pairing with 2 hydrogen bonds, and the figure on the right represents the guanine-cytosine pairing with 3 hydrogen bonds. Very often, a hydrogen atom can jump from one place to another within a molecule. A hydrogen atom is a proton and an electron, and it's the proton or the nucleus that is important here. That proton which holds 2 molecules together, sits closer to one side than the other. In fact, it can sit on either side, but it doesn't want to sit in the middle. It's most comfortable energetically to sit on either sides. And it's known that the proton can sometimes jump across to sit on the other side, but to do that, it has to quantum tunnel. It has to sort of punch through a force field, but it does that occasionally. So from time to time, when these protons quantum tunnel, you get a slightly different form of these nucleotides called the tautomerized form. Now, if the DNA strands split apart where these nucleotides are in the tautomerized forms, when the DNA replicates, these tautomeric bases pair differently. So A doesn't pair with T, it has to pair with a C. So what you get is a mutation, a change in the structure of DNA if this proton tunneling takes place. This was suggested in 1963 by a Swedish physicist by the name of Per-Olof Loudon. A recent research paper on quantum mechanics in DNA determined that the G to C pairing reverse reaction has a significant reverse barrier, while the A to T has a small reverse reaction barrier, indicating that the A to T pairing to tautomer is unstable. The G to C pairing could play a role in spontaneous point mutations provided it can survive long enough to pass through the replication machinery. On the other hand, the A to T, like the likelihood of the tautomeric state being populated via double proton transfer from the canonical state is trivial and is unlikely to be a biologically relevant pathway for spontaneous point mutations. Scientists have discovered that DNA contains a kind of a built-in timer that clocks the frequency with which mutations occur. They show that DNA bases can shapeshift for a thousandth of a second, transiently morphing into alternative states that allow molecules' replication machinery to incorporate the wrong base pairs into its double helix. The researchers claim that these rare mismatches could underlie genetic changes that drive evolution as well as the development of diseases such as cancer. The iconic double helix shows a static double-stranded structure, but it turns out that on rare occasions it can morph into other shapes that exist for an exceptionally small periods of time. We can now correlate quantum tunneling in DNA to the formation of cancer cells. Cells become malignant when mutations in multiple genes that govern proliferation accumulate. Changes in key genes can have a startling impact on cell shape and function. Normal cells have a spherical or solid nucleus, whereas cancer cells have an uneven shape. Cancer cells change shape in order to adapt to different environments. The form of the cell is determined by the tissue in the body. This is important because understanding how and why cell shape changes occur might help doctors identify which cancer genes are active, thus assisting them in treatment selections. As a result, understanding how proton tunneling in the fundamental Watson-Crick base pairing of guanine and cytosine in DNA is a source of mutations and how it might lead to the formation of cancer cells can enable researchers in finding answers to outstanding issues, potentially saving thousands of individuals in Canada and throughout the world. Thank you.